Hi everyone. Um, so before we start with like the actual review, I know some of you guys had questions about like what the exam format is actually like. The format is just like your problem set. So Laura's going to upload the exam just like how she uploads like a Word document for the problem set, and you're going to be filling it out. You're going to be typing directly on the document, exactly like how you do for your problem sets. Um, Correct me if I'm wrong, Rachel, but I think it opens this Saturday at noon and it closes Sunday at noon.
It does, yeah.
Okay, so you have like a full 24 hours. That's 24, right? Yeah. You have
Yeah,
like
and
a full 24 hours.
the exam is designed to take you about 2 to 3 hours, I think, is what at least what it was last semester. I'm sure she probably kept it the same.
Yeah. And do you know if it's like open book? Because I know during the fall it was open book. I mean, there's no way to stop you from using the internet. So essentially it was like open internet, open book, open notes. The only thing that you couldn't do is consult with your friends. You
Yeah,
couldn't
I
ask
think
your
it's the
peers.
same.
Okay.
Because
Yeah,
you still have to turn it into like Turnitin.
right.
So check your answers and stuff if you're too similar to somebody.
Yeah. Um, so the format is a Word document, just like your problem set. You have 24 hours starting at noon on Saturday, the 26th. Um, and you submit it on Turnitin. Does that answer your question, Zev?
Yeah, it does.
Okay. Do you guys have any other questions about like exam logistics before we jump into this?
Well, the first slide is kind of an overview. If you
Oh,
want to go to that one.
yeah. Sorry.
There
Yeah.
you go. No, you're chilling. You're good. Yeah, so this is just the some topics to know, non-exhaustive. And our little disclaimer is we picked the topics that are most important, but we do kind of go over everything. So I think it's pretty extensive. But just some advice is that the exam is open book, so the problems are very likely to be problem solving based. So focus less on memorizing, more on understanding what happens, like especially when you change different parts of like a structure, a mechanism, or a pathway.
Yeah.
Just some
Okay.
tips.
Oh, yeah. You just mentioned this, right?
Yeah.
Okay. Yeah. And again, Rachel and I, we're going to stop periodically and just going over like we'll obviously be asking you questions, but we'll also throw in like some tips about how to approach a different kinds of question. Um, because again, Laura's not a big fan of like questions to just ask you vague rogue memorization. It's more answering a concept than anything, and we'll show you how to approach those. Okay, so this is chemistry of our groups. Do
Yep.
you want to take this one or should I, Rachel?
I can do this one.
Okay, sweet.
Okay, so amino acids. So when we first talk about chemistry, the main thing we're talking about is amino acids. And then this is just a quick overview of the structure of amino acids. So remember you have your amino group, which is the N terminus, and the carboxyl group, which is the C terminus. Every amino acid has this structure, and the variable R group, or the side chain, is the only different aspect of each amino acid. The rest of them have this alpha hydrogen connected to the alpha carbon. You have your carboxyl group in red, amino group in blue. And yeah, like I said, below all amino acids have these components. The backbone runs from the N to C to C to O. That's your backbone. It's just kind of like that middle part. So when we talk about backbones, that's what we're talking about. The R groups give each amino acid its unique chemistry. So there are 20 total amino acids. You don't need to know the specific names, and if she ever mentions one, she'll give you like the name or the like. I mean, it's open books. So honestly, you could look it up too, but she won't ever like ask you to like go in detail with each one. But there are 11 polar and 9 non-polar amino acids, and within the polar group, you have 6 neutral, which are uncharged. So if you ever hear her say polar uncharged, that's what she's talking about. And then polar charged, which either would be acidic or basic. And when they're charged, the basic amino acids are positively charged and the acidic amino acids are negatively charged. And non-polar amino acids don't have a charge. So another important part of the amino acids is their states at physiological pH, because that's what they'll be at in the cell most likely, which was 7.4. So at physiological pH, which is 7.4, the acidic amino acids, like you can see above, will be negatively charged, and the basic amino acids are positively charged. The other amino acids, whether they're polar, uncharged, or nonpolar, don't have a charge. And then just to remind you that nonpolar means hydrophobic and polar means hydrophilic. So peptide bond formation is important because this creates your proteins. Peptide bond formation is a dehydration reaction, which means water is lost, as you can see in the first diagram. When the C and the N come together for the two amino acids, so the C terminus of the first one and the N terminus of the second one, they lose that OH on the carboxyl group and the hydrogen on the amino group, and it leaves as water and creates this bond between the carbon and the nitrogen to connect the two amino acids in a protein or a polypeptide. So basically, just remember that the amino acids are read from the N terminus to the C terminus. And another name for a dehydration reaction is condensation reaction. So if you ever see that on an exam or something, it's condensation and dehydration are the same thing in terms of reactions. And this reaction is catalyzed by the rRNA, so the ribosomal RNA of the ribosome, and it creates a covalent bond. It's a pretty strong bond between the amino acids to hold them together. And if you ever break a peptide bond, this would be a hydrolysis reaction. It's basically just the reverse. Water is put in order to break that bond between the carbonyl group and the amino group. And Sebastian, if you ever want to add anything too, you can totally, but it's up to you. Okay. So when we talk about proteins, we got to know your structures. And I know we talked a lot about this at the beginning of the course, but I'm just going to briefly go over it again because it's very important to remember which types of bonds are involved for each structure, because she likes to ask a lot of questions about that. So the first two levels, you have primary and secondary structure. So primary structure is basically what I just talked about. It's the sequence of amino acids held together by peptide bonds, which are covalent bonds. The only time you're going to really see covalent bonds be talked about in protein structure levels is in primary. There will be a little bit of in disulfide bonds in tertiary, but if you we'll talk about that. But for the most part, covalent bonds will be associated with primary structure. Secondary structure is the formation of alpha helices or beta sheets, which is caused by hydrogen bonding between the amino groups and the carboxylic acid groups. So this is basically just the backbone of the amino acid. So if you remember from the amino acid slide, that backbone from the N to the C, N to C to C, and that's that backbone. That's the only part that's being used. It's just the NCC, NCC pattern. That's the backbone. So basically, the big bond that we want to talk about here is hydrogen bonding. When we talk about hydrogen bonds, this is the most important structure that they're involved in. Hydrogen bonds are non-covalent bonds. And as you can see where Sebastian's question just was, that hydrogen bonds in between the sheets and in between the alpha helices, that's where those hydrogen bonds are, and they're used to stabilize these structures. And another important part is to remember that the backbone, the R groups are not involved in this structure at all. There will be no interactions with the R groups. There's nothing, as you can see on the diagrams, they're just kind of there. So the alpha helices, the R groups are oriented, so they're up and down, above and below the backbone. And then the beta sheets, the R groups are oriented so that they're on the sides of the plane, but they're not involved in any type of bonding or reactions here. And then this was just a quick thing on hydrogen bonds because some people got confused last semester, so we're just going to go over it again. So Professor Francis, she doesn't use the terms hydrogen bond donor or acceptor, but you might see that if you look up research. Basically, the hydrogen bond acceptor is the atom with the partial negative charge, and the hydrogen bond donor is the atom with the partial positive charge. So in this first little diagram, you can see the oxygen is a partial negative and the hydrogen is a partial positive, because that hydrogen is basically what's allowing that bond to form. And just a reminder, hydrogen bonds need to form between a hydrogen and either a fluorine and oxygen or nitrogen. You're never really going to see a fluorine molecule be used in cell biology because you shouldn't really have fluorine in your cells. It's not good. So really in this class, you're only going to see them form between oxygen and nitrogen. So basically the first molecule has to have, wait, can you access? Yeah. The first molecule needs to have the oxygen or the nitrogen, and the oxygen or the nitrogen needs to be bound to a less electronegative atom. So as you can see from any of these, the nitrogen is bound to the hydrogen, which is less electronegative than nitrogen, and carbon, which is also less electronegative. And then the second molecule needs to be where you're getting that hydrogen from, and it needs to be bound to either an oxygen or a nitrogen. So this is just an important takeaway. And then a good way to remember electronegativity is this Fonkel-Berish type thing. But this is just to kind of help people with hydrogen bonds. Some people get confused, but hydrogen bonds, secondary structure. Does anyone have any questions before I continue? You guys good? Okay.
I'm good.
Okay, cool, thanks. All right, so tertiary structure and quaternary structure. Tertiary is the interaction between R groups. Now the R groups are playing a part in bonding to give the overall fold of the single polypeptide. So that will be this first diagram above on the right. And then this is where disulfide bridges come into play. These are covalent bonds, but they're only formed between cysteine molecules, and they help to stabilize tertiary and quaternary structure. So you'll never really find disulfide bridges in secondary or primary. You're only going to find them in tertiary or quaternary. And then quaternary structure, not all proteins have it. Some proteins are just one polypeptide, and you have a tertiary structure. But if it has multiple polypeptides, then they're going to fold in on each other and create this quaternary structure. So I think this is hemoglobin. Yeah, that's hemoglobin. So there's 4 polypeptides, so you can see them in the different colors, and they're all bound, which is or interacting, which makes this quaternary structure. So then just a quick question for you guys. What effect would a molecule that disrupts hydrogen bonding, such as urea, have on protein structure? What do you guys think?
Well, I would assume that we couldn't have secondary or tertiary or quaternary structures.
Mhm.
Maybe,
Yep.
yeah, maybe some
Mhm.
tertiary and quaternary through like ionic bonding or other types of electrostatic bonding, but
Yeah.
definitely no secondary structures.
Exactly. So it will definitely disrupt secondary and unfold the alpha helices and beta sheets. And it could, you're right, it could affect tertiary and quaternary. It doesn't have to, but it could if like they're relying on the hydrogen bonding. But it definitely would not affect primary structure. So, good job. And then we have one other question. Which of the following may be considered an example of tertiary protein structure? Van der Waals interactions between 2 phenylalanine R groups located far apart on a polypeptide, hydrogen bonds between backbone amino and carboxyl groups, covalent disulfide bonds between cysteine residues located far apart on a polypeptide. So which of these would be considered?
Wouldn't 3 be considered the tertiary protein structure?
There's one more.
One as well.
Yep, good job. So yeah, they're both example of tertiary structure because for 3, yep, disulfide bonds are an example of tertiary structure. And for 1, van der Waals on 2 amino acids located far apart is an example of how proteins fold in on each other. So that would be 1 and 3 would be your answer here, and 2 would be secondary structure. Good job. Sebastian, do you want to?
Yeah. Okay. What are we talking about? Okay. Domains and subunits. So the way I see it is a domain is essentially a part of a protein that has some kind of function. Like the definition says, it's independently folded section of a single polypeptide. So I could explain this. Let's look at this green structure for a second. I I have no idea like what this protein is, but let's say somewhere in this protein, there's like a domain. Like let's say this region is a domain, and that domain is responsible for cleaving covalent bonds or something like that. That domain of this protein is particular for cleaving covalent bonds, but maybe it has another domain that's responsible for promoting or for synthesizing covalent bonds. So a single, I may have just given this question away, but what do you guys think about this one? A protein with one polypeptide chain can contain multiple domains. What do you think?
Yes.
Yeah, it definitely can. So again, let's ignore like this red part for a second. A domain is just a part of the protein that folds independently and has some kind of particular function. A single polypeptide chain can have multiple domains. Domains are usually evolutionary conserved, which in simple terms means even through several years, thousands of years even of evolution, that particular section, that domain stays conserved. So even if this cell goes through, hundreds of years of evolution, that particular region of the protein will stay evolutionarily conserved. Subunits are only relevant when you're talking about quaternary structures. A subunit is just one polypeptide chain in a quaternary structure. So like Rachel just said, a quaternary structure is a protein with multiple polypeptide chains. So this is hemoglobin. It has 4 different subunits. Each color, or like each colored ribbon, I guess, represents one polypeptide chain. Like this is one polypeptide chain. Maybe this is some protein and it's doing its own thing. Sometimes it's not uncommon for more than one polypeptide chains to come together to do something else. That's called the quaternary structure, like this one. So a subunit is just one polypeptide chain in a quaternary structure. So for instance, this would have 4 subunits because there are 4 different polypeptide chains. Okay, so any questions about domains or subunits? Okay, there are different ways that you can model your protein. Each of these are different examples that you'll probably see on like Google Images or on a computer for how to visualize your protein. And each of these have their own pros and cons, which I think we talk about. Oh, I guess not. Each of these have their own pros and cons. So for instance, with the ball and stick model, I believe this one does show you R groups. Yeah, okay, awesome. It shows you all the atoms involved in your protein as well as the R groups. So with that, you can you could see like the relative size of your protein. You could see where the R groups are positioned, so you can estimate what kind of interactions it's most likely going to have if you know which R groups you're dealing with. The ribbon model is really good for showing you secondary structure because as you can see, there's all these helices everywhere. You could see some of the beta sheets in the back. So the ribbon model is good for illustrating secondary structure. Backbone is good for primary structure. This space fill is good if you want to see how much space your protein provides. It shows you the van der Waals radius for all of the atoms. So if you want to see just how much space or how bulky your protein looks, the space fill model would be good for you to use. Definitely know the pros and cons for each of them. If you want a little more detail on those, I believe our review, week 1-1 review, that PowerPoint and that video has a pretty exhaustive list of all the pros and cons. You might see a question like that on the exam, like Laura may ask something like, here's a protein, which model is the best one if I want to see. Oh, okay, so the question might be like, let's say I want to see the R groups for this protein, what model should I use? And in that case, if you want to see the R groups, you should probably use the ball and stick model. That's very good for R groups. Whereas ribbon, you can't see the R groups in ribbon, so this is one that you probably want to stay away from. Usually things to that effect. Do you guys have any questions about modeling?
No.
Okay. Okay, sweet. So like Rachel mentioned, what distinguishes each amino acid from each other is the chemistry of their R groups. So they all have the same backbone, but what makes them different is their R group, their side chain. primary structure is all about just building the polypeptide chain. That is exclusive to covalent bonds. Secondary structure is exclusive to just hydrogen bonds. Hydrogen bonds with the backbone, not the side chain. When you get into tertiary structure, excuse me, oh geez. When you get into tertiary structure, that's where things get a little more complicated, only because in tertiary and quaternary structure, that's when you have to start considering your R group interactions. So notice that R groups have different chemistries. You have your nonpolar, polar, and you have your polar charged ones. So depending on which ones are coming together will determine the strength of that bond. It'll determine how they're interacting. So for instance, if you have two nonpolar amino acids coming together or pointing at each other, that would be, because they're each nonpolar, that would be a van der Waals interaction. Let's say you have two polar molecules coming together. Because they're each polar, you would have a hydrogen bond. If two oppositely charged amino acids came together, you'll have an ionic bond. We have a slide that kind of runs down like all the different interactions that you'll go with. But yeah, definitely this is probably a chart that you'll probably need for this exam. Okay, do you want to add anything, Rachel? Or do you guys have any questions about this? We have questions for you soon.
Could you elaborate on the van der Waals interactions for the nonpolar?
Yeah, so a van der Waals interaction, it's also called like London dispersion forces or induced dipole. There's a bunch of names for it, but essentially it's when you're trying to put two things together that don't have like a natural attraction to each other. So for instance, if you're trying to put two polar amino acids together, let's like just focus on the tyrosine and the asparagine. The tyrosine has this oxygen that's very electron rich, so it's partially negative. Let's also consider like maybe the hydrogen on this nitrogen right here. I know it's a little small, but hydrogen is of course pretty electron deficient, so it's partially positive. So when you put these two together, you're essentially interacting the partially negative oxygen with the partially positive hydrogen. And if you've ever like played with magnets together, you know that when you put two magnets together, you put the minus end with the plus end, and that causes the magnets to like stick. That's kind of how this would work. That's what a hydrogen bond is. But with nonpolar, because these amino acids are nonpolar, they don't have any partial negative, partial positive. They don't have that kind of dipole, hence nonpolar. So the only form of interaction they could have is a van der Waals interaction. To put it very plainly, it's just electrons are swirling around each of these atoms. Obviously, like they're always rotating, and at some point you're going to get the electrons concentrated at like one particular side of the atom. So it creates what's called like a partial dipole, or sometimes an induced dipole. So it's that temporary moment where the electrons have aligned themselves in a position where they make like a fake dipole or a temporary dipole. And that temporary dipole is what allows them to bond together, hence making a van der Waals interaction. It's a very, very weak interaction because, again, there's no natural polarity like there is with polar amino acids. So it's a very, very weak kind of bond, but you still can get some interaction with them. I'm sorry, that was like a huge explanation for this. But does that answer your question, Zev?
Yes, it does. Thanks.
Okay.
I had a question on top of that. So how does that relate to the space filling model that you were saying? Because it shows the van der Waals radiuses.
Yeah.
So what exactly is it showing there of the bond?
The space filling one?
Yeah.
It shows you like the radius, okay, so you know how atoms have electron clouds that just kind of like circle the perimeter of the nucleus?
Yeah.
That's essentially like what this space filling shows you. It shows you like the radius of the electron cloud. So these atoms are all like relatively the same size, but let's say I were to insert like iodine or just another massive atom in there, rubidium, I think, whatever, that would probably have a much larger, in the spacefold model, you would probably see a much larger atom. So if I try to put like iodine somewhere in here, iodine is a huge atom compared to like all these carbons and hydrogens or whatever. So it's kind of hard to see the difference in sizes with this. But again, if I put like another larger atom, you will definitely see the difference between that very large atom that I've put right here and the rest of these atoms that are relatively more or less like the same size, kind of. Does that answer your question?
Yeah, thank you.
Yeah, no problem. Okay. Steric hindrance. Oh, okay. So there's a really, I don't know if we, I can't find it. There's a really popular question. You guys might have like probably seen it in like a problem set or something, but a really popular question that Laura likes is, let's say you have some substrate, you have some protein, and it's going into a protein pocket, and you have to determine, okay, this protein inside this pocket, you have to determine like what kinds of bonds are going on. So if you mutate a proline, a non-polar amino acid, into a polar amino acid, then you have to determine like what bond you're forming now, since you mutated this into this. So that's one thing to bear in mind, the chemistry. But another thing to bear in mind is the steric hindrance, or the bulkiness of the amino acid. So notice that all of these, the glycine, proline, phenylalanine, these are all non-polar.
Phenylalanine.
Thank you. Phenylalanine, whatever. All these guys have the same chemistry. They're all non-polar amino acids. So let's say you get a question where it's like, a protein is binding into this pocket, and the protein has this amino acid pointing towards this amino acid inside the pocket. So what interaction would you get? You would expect a van der Waal interaction, because these are both non-polar. But now let's say the question mutates the proline to phenylalanine. Well, they're both non-polar. So your first thought may be like, oh, okay, maybe there's no difference. However, it's good that you consider the chemistry. You notice that they're both non-polar. So as far as the chemistry is concerned, you're not going to have like a difference in the bond chemistry, but what you will have a difference in is the size. So notice that your proline compared to, I'm not even going to try anymore. The proline compared to this benzene ring is humongous. It's taking a lot more space than this is. So if you mutate the proline to this amino acid with the giant benzene ring, it actually will affect the interaction between your protein and your pocket. Because you're switching it into something that's much, much larger, there's a whole lot of steric hindrance. There's a whole lot of bulkiness there. So it is actually tarnishing that interaction between the glycine and phenylalanine. Did I get it?
Phenylalanine.
Whatever. It's a problem.
Yeah,
for another day.
And just one thing to add, just kind of side note, like you'll never find like a proline or a phenylalanine like in the cell membrane, like in like the hydrophobic portion of it. You'll never find them there because they're too bulky and they stick out. But you'll find a lot of glycine. So that's just like one way you can also think of it.
Oh, okay, yeah. This again, this kind of like summarized what I was saying. I think. Wait, tyrosine, very similar.
Yeah.
Okay, yeah. So
and then practice question is basically what I just said.
where would you find these amino acids? Oh, okay. I mean, yeah. We'll let's just do it all over again. Why not? So again, like I mentioned, a question that you're most likely to encounter in the exam is like, let's say this amino acid is trying to interact with this one. Well, you have a polar uncharged coming with a non-polar one. So I know in my Chem 112 class, we called this a, what do we call this? An induced, no.
Did you guys just lose him too?
Dipole induced.
Yeah.
Oh, can you guys hear me? Can you hear me now? You can?
Now we can hear you.
Oh, okay. I was like, damn. Okay, word. Oh yeah, I just got a message that my internet's a little unstable. Sorry about that. If I ever get cut off, feel free to like let me know or interrupt me or drop it in the chat. I'll try to fix that. What was I saying? Oh, this kind of interaction. Okay. I know in chemistry we called it a dipole induced dipole because you have a dipole molecule coming together with something that requires an induced dipole. I don't know what Laura's terminology would be for like these kinds of interactions. I think it was van der Waal. I think she just classified it as van der Waal. I'm not too sure. But for instance, if you have a question where you mutate this one to the cysteine, again, even though you're changing non-polar for a non-polar, you should consider the steric hindrance. You should consider the size of these amino acids. Okay, so this practice question, where would you find these amino acids in a folded protein? Okay, where would you find this amino acid in a tertiary folded, completely folded protein? On the interior or exterior?
I mean, assuming that the exterior is where is exposed to solution, it would be on the exterior.
Yeah, yeah, exactly. And that's a good call. Most solutions, unless like stated otherwise, will be an aqueous solution. It will be a polar solvent. Again, the question would have to like specify otherwise. It would have to specifically say you're submerging your protein in a non-polar solvent. I've never seen a question like that, but that would be pretty cool.
I
In
was
that case, yeah,
thinking-
it would be inverted.
I was thinking about like the beta barrel example,
The beta barrel.
where yeah, she talked about like the specific transmembrane protein, where the inside would
Oh.
be hydrophilic.
Yes, that's right. That's right. I remember. Okay, yeah. So to clarify, yes, your solvent is polar. So you're totally right. You would find the tyrosine, the polar amino acid on the exterior. And the non-polar, like you just said, by extension, you'd find those on the interior. Again, because non-polar substances want to stay away from polar substances since they don't mix. So this amino acid will most likely be found on like the inside. It'll be tucked away somewhere in the interior. Okay, any questions about these? Here's a cool little bond summary about most of the bonds we talked about. So your covalent bonds are peptide bonds. They hold the backbone together of amino acids. Disulfide bonds, they only form in tertiary and quaternary structure, and they need 2 cysteine amino acids, right? Yeah, you need 2 of these to form your disulfide bridge. The non-covalent bonds are ionic bonds. Here are some of the instances where you will get an ionic bond. Hydrogen bonds are your polar uncharged coming together with polar uncharged. You can see the rest. So covalent bonds are much stronger than your non-covalent bonds, and here's what they look like. Primary structure, these are the bonds and the interactions that you'll get. These are the interactions and bonds that you'll see in secondary, and these are the bonds and interactions that you'll see in tertiary and quaternary structure. Okay, any questions about this?
I'm all set.
Sweet. Okay, so true or false, in an alpha helix, the R groups of the amino acids in the alpha helix are available to make interactions with other parts of the protein and or other molecules. What do you think? I don't know the answer. I'm just kidding, I do.
I would think they'd be able to.
Why do you say so? Why?
I mean,
It's so aggressive.
because they're there, and I mean, I would assume, I would just assume that they would.
Yeah, that's fair.
Because they're,
Is it the way they're folding and like the hydrophilic interacting with the hydrophobic?
Wait, I'm sorry. Could you repeat that, please?
Is it like the way that they're folding and the hydrophilic is interacting with the hydrophobic, or is that not? Is that just for like the beta barrels?
So the hydrophilic and the hydrophobic interactions. Do you mean like the R groups interaction?
Yeah.
Yeah. Oh. Okay. I see.
that's what makes tertiary structure, right? So
Yes.
we can
yes.
have an alpha helix that's a part of a tertiary structure.
No, the alpha helix is part of secondary structures. But okay,
Yeah.
I'll clarify this question. You were right. Alpha helices can make interactions with other R groups or other molecules. But just to quickly clarify, in secondary structures, in alpha helices or beta sheets, the only interactions that you're going to see are hydrogen bonds. So secondary structures like alpha helices or beta structures, those are exclusive to hydrogen bonds. There's no other interactions there. But with R groups, you can get a whole mess of interactions. So the reason that you still can get R group interactions with other proteins, other molecules in alpha helices is because the R groups in alpha helices stick out from the core of the helix. So if you have like your spiral right here, those R groups are sticking out. because again, what makes the structure of your alpha helix is just between the backbone. It is not involved the side chain. So because your side chain is sticking out, because your R group is sticking out, they are available to make interactions with other things. Because, so the answer to why is because they're sticking away from the helix core. Do you guys have any questions about this? I'm sorry if this got like a little confusing.
No, that makes sense.
Okay, word. All right, and if you mutate the protein of interest at the 21st amino acid, have you mutated the amino acid closest to the N or the C terminus? What do you guys think? How long is this protein?
Nope, that's part of it.
Well,
I would assume the N terminus because most proteins are very long, and so that would be closer to the beginning.
Okay.
Yeah.
Okay,
That's right.
that's a good call.
So for this one, pretty much this is one of her questions too. So she doesn't specify because she wants you to know that if there's no specification, you should assume that all proteins are at least 100 amino acids long.
I actually don't know that.
Well, now you do.
We'll learn something new every day. Sweet. Okay, more questions for you. So, two different proteins have the same domain in the structure. Which of the following is true? So I'll give you a second to like read these. Feel free to just like shout it out when you think. And again, like if you're honestly not sure, just take like a swing at it. You know, that's the whole point of these reviews.
Wouldn't it be D because the domain is only present in the quaternary structure?
I think that's subunits. I think it may be subunits that you're thinking of.
Okay.
But yes, subunits are specific to quaternary structures.
So would that be B?
Yep.
What do you
Yeah,
think?
it's B. But yeah, why? Why do you think it's B?
Because they a domain is the specific way it's folding in the tertiary structure, and so that means that the R groups, if they have the same domain, it means that those amino acids have to be the same for that specific domain, but it doesn't mean that the entire proteins are the same.
Perfect.
Mhm. Yeah, that's
Stuff.
that's perfect.
Yeah. It's not A, because if you had the same primary structure, you would have the same protein, right? But here you're dealing with two different proteins. Even though they're different, they could have the same domain, but if they have the same domain, like you said, that domain is specific to a certain thing. So at least some part of those two different proteins must have a similar structure to have the same domain. So that's true. Okay. I think we answered this one. Yeah, we answered this one before. One polypeptide chain can have multiple domains. Okay, you have a protein that has a mutation that changes the aspartic acid on the 12th amino acid in the polypeptide sequence to a glycine. So aspartic acid, that is a positively charged amino acid. And this is a, polar positively charged amino acid. And this is a non-polar amino acid. So you're going from polar positive charge to non-polar. Does this mutation impact primary structure? Oh, yeah, these are very good. Yeah, these are questions this woman loves. So what do you think? Do you think that's going to affect the primary structure?
By definition, it does.
Definitely. Yeah. If you mutate anything of the amino acid, if you mutate the 12th, the first, the 200th amino acid, you're affecting the primary structure. You're affecting the polypeptide chain. So definitely. What about secondary and tertiary structures? Let's start with secondary. What do you think about secondary?
I don't think so.
Why do you say that?
Because the R groups are different, but the secondary structure is due to hydrogen bonding from the backbone, and the backbone of all amino acids is the same. I
Exactly.
don't know if that makes sense. Yeah.
Yeah, no, that's totally right. Secondary structure only involves the backbone of your amino acid. Here, you're not changing the backbone. Obviously, you're just changing the side chain. So yeah, your secondary structure should remain fine. How about tertiary and quaternary?
Sarah, I don't mean to take up all like the answering time if you want to say something.
No,
It's
you're
kinda
fine. I'm just
hard
I'm
without-
looking through my notes right now.
Okay.
We're so considerate. Wow.
It's like I always feel so self-conscious.
Yeah, no, you're fine.
But for tertiary and quaternary, I would say yes, because we're going from a non-polar to a negatively charged, and so the negatively charged aspartic acid can make ionic bonds. It could make hydrogen bonds.
Exactly.
So that could change the folding.
Exactly. Tertiary and quaternary structures, those do consider the R group. So if you mutate that R group, especially if you go from something dramatic like a polar positive charge to a non-polar, yeah, you're most likely gonna seriously affect the chemistry of that and the folding. So tertiary and quaternary, I do suspect would be affected in some way.
So could I ask a quick question? Kind of.
Okay, what's
So,
up?
so for the backbone, creating the secondary structure through hydrogen bonding,
Mhm.
I'm a bit confused then as to why it would sometimes go into beta sheets and why at other times it would go into alpha helices, because if changing the amino acids doesn't change the secondary structure, then what dictates whether it goes into a beta sheet or a alpha helix?
That's a good question. Do you have a Rachel? Do you want to explain that one?
I'm so sorry. I was looking at my phone.
No, you're fine.
Yeah. Wait, which one are we talking about?
It was so he asked why so in secondary structure
Yeah.
only the backbone is involved in secondary structure, so
Mhm.
what like will determine whether you get an alpha helix or a beta sheet, and if I remember correctly it's about like the position. So
Yeah, it's
alpha heli-
positioning.
yeah, alpha helices will make those hydrogen bonds every fourth amino acid.
Yeah.
Yeah, whereas beta sheets, they happen more frequently. I think, stop me if I'm wrong, but I think with beta sheets, it's like every 2 or 3 amino acids will fold up to make that beta sheet. But with alpha helices, it's like every 4th or 6th um, amino acid backbone, right?
Yep.
Okay. Actually, never mind. I actually had a dream about secondary structures, but that's
No, you didn't. No, you didn't.
Laura was actually in it too.
No, you didn't.
She didn't
Stop
yell at me, but she gave me that disappointed face. She was like, "Wow, you don't know what's stronger, alpha helices."
Stop it.
Do you know actually which one is stronger?
Alpha or beta?
Mhm.
I don't know. I would think maybe beta, but
It
I actually
is
don't really
beta.
know.
It
It
is
is?
beta, and it's because of the positioning. Like you
That's
just said, Zev, it's about like the positioning of where the hydrogen bonds take place. I remember very clearly. Laura looked at me in the eyes, and she was like, "Wow, you don't know which one's stronger." I was like, "Oh my god." It's getting hot. Protein unfolding, misfolding. I don't know this section.
okay. So this is me. Okay. Guys, I swear we're good TAs. Okay. So what are we talking about? Protein unfolding and misfolding. So these are some of the main ideas. There's certain things that impact protein folding and function, including temperature, pH, salt concentrations, and light energy, like wavelengths and all that stuff. Temperature is your thermal energy. So it's important that the cell can regulate these factors. So because they have an impact on non-covalent bonds. So pH, if you hear pH, salt concentration, and temperature, just remember that they impact non-covalent bonds, not covalent bonds. Covalent bonds are much stronger, and in the terms of this class, we consider covalent bonds to be the strongest bonds to exist between molecules. Just for this class, that's actually not true, but in terms of what we're learning, it is. So covalent bonds need like very extreme conditions to be disrupted. So when we hear about environmental impacts on protein folding and function, it's just going to be impacts on non-covalent bonds. So basically what happens is sometimes proteins can fold abnormally when the cell does not regulate these correctly, and like there's abnormal breaking of bonds and then new bonds form. And when proteins fold abnormally, it can cause aggregation, and it can cause certain degenerative diseases like mad cow disease or Alzheimer's. But and then some terminology. So when you hear denature, that means a protein is unfolding, and when you hear renature, that means the protein is refolding. So having a protein that is misfolded or unfolding can be harmful to the cell. Like I said, it can cause certain problems within the cell. And if it's not corrected, it can cause diseases. So the cell has a response to fix this, which is called unfolded protein response, which we'll talk about in like a few slides. So yeah, so resolving unfolded and misfolded proteins. Cells have certain mechanisms that can respond to the problem of protein misfolding. There's the heat shock response or the unfolded protein response. So the heat shock response will occur in the cytosol or the nucleus. And this occurs when proteins are sent to the proteasome in a certain stress response pathway that the proteasome just degrades them. And then the unfolded protein response is very similar, but it occurs in the endomembrane system. And it's an endoplasmic reticulum specific stress response. And the only difference between these two really is that the unfolded protein response just has to send the protein out of the endomembrane system into the cytosol so that it can be sent to the proteasome, because the endomembrane system and the ER don't have proteasomes. There's also homeostasis mechanisms, but you don't really need to know that much about it. And like I said, they can aggregate and cause these diseases such as mad cow disease and Alzheimer's. Prions, she stressed prions a lot when I was in her class, and then she kind of stopped doing that after. But basically it's just misfolded proteins that are actually infectious and can spread to other proteins. So it's like you have this misfolded protein, it can like cause the protein next to it to also fold incorrectly, and then it can kind of like spread. So it's really not good. And they don't really have a cure for it. They don't know how to fix it. So prions are very bad. And they aggregate because they're folded incorrectly, and they'll hide whatever hydrophobic exposed portions they have. And then this causes the protein to become insoluble, so it can't dissolve in anything. And that's how they stack up on each other and cause these problems within the cell.
Actually had this problem in my lab.
Really?
Yeah.
Oh. All right. So chaperone proteins are special proteins that are recruited to help proteins fold correctly, and then also stabilizes them when they're folding or misfolding. So I like to think of just chaperones like chaperones on a field trip, making sure like the rowdy kids behave. And then this little diagram shows how they kind of stabilize a protein and help it fold in the correct response. Because without chaperones, you're going to have higher amounts of incorrectly folded proteins. So one of the first responses that I mentioned is the heat shock response. So the cell senses that something's wrong with the protein that is misfolded or unfolded. So what it does is basically it just ships them off to the proteasome where it gets destroyed. During the heat shock response, chaperone production is upregulated. So you're going to have more chaperone proteins around in the cell to help either try and refold the protein or stabilize it, or send them to the proteasome where they will get degraded. And then this response occurs in proteins that are misfolded in the cytosol or the nucleus. So that's one important distinction, because then on the next slide, we have the unfolded protein response. Yeah. And this occurs in that endomembrane system, like I mentioned. It's basically the same thing. The cells sense the misfolded proteins in the ER. And as a result, it responds by making more chaperones as well. Except the big difference between the UPR and the heat shock response is that the UPR occurs in the ER and the heat shock response occurs in the cytosol and the nucleus. So some mechanisms that the cell does to either send the protein to the proteasome or kind of just stop production of misfolded proteins is it decreases translation. This reduces the folding load in the ER. So if you're decreasing translation, you're not going to be producing proteins as quickly. And basically the ER can just deal with the proteins that are misfolded first, and then it can go back to translating proteins. But if you're making too many proteins and you have like a few that are misfolded or unfolded, it will just like be too much at once, and it won't be able to like pick out the unfolded proteins or the misfolded ones. The cell also will upregulate chaperone production, like I mentioned, because it will help to refold the proteins that are not folded correctly. It increases lipid biosynthesis. This is for the ER to make more space for the proteins to kind of either refold or for chaperone proteins to take up space. Because if you don't have any space, cells not be able to do anything. So the increased lipid biosynthesis just makes that more space. And then the last part, this isn't in that order, but this part is always last, is that the proteins are sent out of the ER to the proteasome where they get degraded because there's no proteasome in the endomembrane system. So that's the UPR and the heat shock response. So some practice questions. Where in the cell is the unfolded protein response initiated?
That's the cytosol and the nucleus, right?
This one's the ER. So the
Oh,
UPR
yeah.
is in the ER. Yeah. The heat shock response will be the nucleus and the cytosol. So UPR is in the ER. This is how I like to think of it. Okay, number 2, which of the following are ways that the cell responds to unfolded or misfolded proteins? So basically kind of true or false for A, B, C, and D. So for A, does the cell upregulate translation during an unfolded or misfolded protein response?
Doesn't it decrease?
Yep, and it decreases why? What do you think?
Is it just to like reduce the amount that the ER has to deal with?
Yep, exactly. And yeah, so it just decreases the amount because it's trying to focus on what it needs to fix first before it makes more. For B, does the cell increase chaperone production?
Yeah, just the more chaperones there are, like they can refold simultaneously.
Good job. And then for C, does the cell translocate proteins to the ER?
Yes, I'm not sure.
Yeah, so it will translocate proteins out of the ER. I should probably write like into the ER. That would probably be better. But yeah, so this one would be you'd want to go out of the ER because there are no proteasomes located in the ER. So you need to send the misfolded proteins out of the ER to the cytosol where the proteasomes are. Does that make sense?
Mhm.
Okay. And then letter D, does the cell increase lipid biosynthesis? So lipid biosynthesis is what?
That's a part of the unfolded protein response.
Yeah, so the increase in lipid biosynthesis would occur because this creates more space within the ER for the protein response to occur, basically. Good job. And we have our little puppy break.
Did
Usually-
Jason make those?
Of course, well, I made this one. Jason did the cat
Oh.
break.
Okay.
Do you guys have any questions on anything really that we've gone over so far?
No,
No.
I'm good.
I appreciate the puppy break though.
Yeah, isn't it fun?
It was
Okay.
nice, yeah. Just kidding.
One of my best friends just got a puppy today, and I'm actually really jealous. So, okay, I'm doing this one, right, Sebastian?
Oh, wait, sorry.
But which one of us is doing this one?
Wait, I'm sorry. You're breaking up a little bit. What did you say?
I said, am I doing the protein ligand and enzymes?
Let me check this out. Okay, I feel pretty good with this one. Up until like Drake. Wait, what is he talking about? Yeah, you could do Drake. But I got
Okay,
this
I'll do
one.
Drake.
Okay, word. Protein ligand interaction. Wait a minute. Did you guys have heard of like KD, right? This was mentioned in the videos.
Yeah.
Okay, sweet. Okay, so let's see. What am I talking about? So a protein ligand interaction, this happens pretty common within the cell. You have a ligand, you have like some substrate that's going to find its way into a protein, usually like an enzyme, and something is going to happen to the substrate when it's inside this enzyme pocket. This is actually what I was trying to say before with words that I feel like I did very poorly, but this is the kind of question that you would see in Laura's exams. I think I've seen at least one in every exam where you have some substrate and it's like trying to fit into this protein pocket. And right here, it'll tell you there's a glycine, there's an aspartic acid, there's a leucine, whatever. And on the pocket, it would show you like other amino acids, and you have to determine like what kind of interaction will this lysine and this glycine make or whatever. Sometimes the question might mutate it, so it'll be like this got mutated from a non-polar amino acid to a polar amino acid. What happens to the KD? Stuff like that. So a ligand is just any substrate that binds into like a pocket. Here's your substrate or your enzyme. Sorry, not your substrate. This is your enzyme. So for this to work, your protein needs to be, or your ligand needs to be oriented correctly for it to fit inside. It has to have like a good affinity. So it has to have a natural attraction to this pocket for it. So it needs to be oriented correctly and it needs to have a good affinity. So if all of these proteins were lined with nonpolar and all of these were lined with nonpolar amino acids, you're not really going to have a good affinity. Because of course the only interaction that nonpolar and another nonpolar amino acid can make is a van der Waal interaction, and that interaction is very, very weak. So if this substrate is like completely filled with, let's just say hypothetically, it's completely filled with nonpolar amino acids, and this is filled with nonpolar amino acids, you're gonna have a pretty low affinity and a pretty high KD. And I'll talk about KD in a minute. So here's some things that affinity depends on: the number of bonds between the protein and ligands and the strengths of the bonds. So like I was saying before, if you're lined with like a bunch of non-polar amino acids all over the place, the affinity is going to be pretty weak. But if this binding site, if this pocket had a bunch of polar positively charged amino acids, and this substrate had a bunch of polar negatively charged amino acids, well, then you're going to get a really high affinity. Because you have polar amino acids with opposite charges. That's going to make an ionic bond, and those are relatively strong non-covalent bonds. So in that case, this ligand and this protein would have a good affinity, and it would have a low KD. Okay, so let's talk about what KD is. In summary, it's just an equilibrium value. You've probably seen this in Chem 112 several times. The way I always remember KD is the constant of dissociation. This D means dissociation. So if you have a high KD, you have high dissociation. If you have a low number, if you have a low KD, you have low dissociation. So that's always how I remember that. Okay, any questions about this so far?
No.
Okay, sweet. So what are enzymes? What do? They do a few different things. The one that you've probably heard several times is like they increase the rate of reaction by lowering activation energy. So what an enzyme does is it essentially just makes a reaction happen faster. That's one thing that it could do. So for instance, the RNA polymerase is it's an enzyme because you're catalyzing the rate at which these nucleotides could be added onto your RNA template. Those nucleotides could just diffuse in there, but obviously that's going to take forever. So the RNA polymerase actually catalyzes that reaction. It catalyzes how those nucleotides get placed onto the RNA for like synthesis or repair, whatever. So one thing that it does is lower the activation energy. It could make or break covalent bonds, like I was mentioning earlier. Again, that protein must have like a domain responsible for that. Because it is making and breaking covalent bonds, because it has like some chemical reactions, that's what classifies that protein as not just a protein, but an enzyme. They're needed because, like I just mentioned, they speed up the rates of reactions that usually happen very slowly. RANGAP is another one. So RANGAP shovels out a GDP to allow GTP to come in its place. That reaction can happen on its own. The GDP could diffuse on its own, but like I mentioned before, that's going to take a long, long time. For GDP to just shovel itself out, that's going to take way too long. So there are enzymes like RANGAP that's responsible for not waiting for the protein to get rid of GDP, it shovels it out for it. So again, it just speeds up the rate of whatever reaction. You could also regulate proteins this way. Not all enzymes are made 100% for the proteins. Don't really know exactly what that means. I guess that's like, it's non-specific, and you could correct me, Rachel, if I'm wrong, but this is referring to like not all enzymes are like 100% for like one protein. They could be used for like a more broad range of proteins.
I think what it means is that the majority of enzymes are made of proteins, but there are some called just that are RNA and protein mixture, which are the ribozymes. I think that's just what that one's talking about.
Oh, like rRNA.
Yeah. So
Okay,
I think,
got you.
yeah.
Okay, that makes sense. Okay, do you guys have any questions about the different functions that enzymes have?
No.
Okay.
I have a question. I'm kidding. I don't have a question.
Yeah.
No, I'm kidding. I don't have a question.
Oh, God, I thought that was Sarah.
Oh.
My bad. Reaction coordinate. Okay, yeah, you've definitely seen this hill like a million times. What an enzyme does is, again, it just lowers the activation energy. This is how much activation energy is needed for the reaction to actually proceed. An enzyme just lowers that, so you don't need as much energy for that reaction to proceed. Spontaneous reaction, energy product is less. Energy product is less than what's released.
This is basically a Gen Chem review.
Yeah, I remember this as exothermic, like when you end up with less energy than what you started. I haven't
Yeah.
seen it written in these terms, but I'm sure the concept is the same. So spontaneous or an exothermic reaction is just where your products have less energy than what you started with. Non-spontaneous or endothermic is when your products have more energy. So if the hill ended like somewhere over here, if your products ended up with more energy than they started. Okay, that does make sense. Okay.
That's on the MCAT also.
This?
Yeah.
I bet, yeah.
Mhm.
This is another way for enzymes and how they work. This red and blue tubes here, these are all your substrates. This green pocket thing is your enzyme. So one thing that an enzyme could do, again, an enzyme is just something that catalyzes a reaction. So it makes a reaction happen faster. And one way that it could do that is it could squish like two different proteins together, and forcing them, constraining them to be in this close proximity can encourage the reaction to happen. Another way that an enzyme could work is it could just rearrange the electrons of your substrate. By rearranging the electrons, you're rearranging like the connectivity, you're rearranging like what this protein can bind to, what it can't bind to. So that's another way that it could speed up a reaction. An enzyme could also force the substrate into like a particular conformation. So as you guys know, the shape of a protein determines its function. An enzyme, if it needed a protein to quickly change shape to do a different function, it could just force it into this kind of conformation to, again, favor some kind of reaction. Okay, so here's a question for you. If during an enzyme catalyzed reaction, an intermediate forms in which the substrate is covalently linked to the enzyme via a serine residue. Can this occur at any serine residue, or must it occur at a specific one? So
This is an MCAT question also.
Yeah.
I literally took it from the MCAT.
Oh gosh. If I could like simplify the language a little bit.
Yeah. Simplify it.
Yeah, so if you have a substrate, this red thing, so let's say a substrate is finding an enzyme, this green enzyme, and that enzyme goes inside and it finds a spot where it could covalently link to the substrate could covalently, remember that's a strong bond, it could covalently link to the enzyme. Do you think it could happen at like any spot where there's a serine residue, or do you think it must happen at a specific spot where there's a serine residue?
What is a serine residue?
It's just,
Yeah,
sorry, go ahead.
No, you could probably answer better than I can.
It's just it's just like the amino, it's just a serine amino acid. Like a residue is just like a amino acid by itself, basically. So it's just a serine amino acid.
Okay.
If they all have the same structure, wouldn't they be able to?
So the main, like, I guess the simplified version of this question is, are enzymes specific to their substrates, or can they do it to similar substrates? That's basically the simplified version of this question. I just put it in MCAT terms to like, because I guess it kind of shows you like the crazy wording, because Laura sometimes does that on hers. So basically, the short answer to this is that enzymes are very specific. So the reaction would have to occur in the terms of this answer, that it would have to occur at the specific serine residue. It can't just bind to any residue, any serine residue or any serine amino acid. So the main takeaway really from this question is that enzymes are very specific and they can't just bind to any substrate. It has to be a specific substrate. Does that make sense? I know it's kind of worded strangely, but if you guys are pre-med, that's what you're looking at for the MCAT. So
So
Yeah, that makes sense.
okay.
yeah, okay, cool.
Word.
I'm also never taking the MCAT, so
You're lucky.
Yeah, either.
There's like, oh no. Okay. Yeah,
Okay, sick. All right, so then you guys are chilling.
I don't got this.
Oh, this one's me. What do you mean? ATP World Tour.
Oh,
You see it?
yeah. I didn't see this. Okay.
I don't, Jason put this one in. All right.
Clown.
So protein function regulation. Go over this. This one's pretty simple. Basically, you don't really need to know a lot about allostery. You just have to know that allosteric binding means that something is binding to the enzyme at a spot that is not the active site, so not at that site we were talking about before, and that it's a form of regulation. So you have allosteric inhibition and allosteric activation. So if you look at this first set of regulation, which is the inhibition, you have an enzyme, you have the active site, and you have this green substrate, like this semicircle. Basically what it does is this inhibitor, which is this red square, will bind to the allosteric site, which is not the active site. It's just another spot on the enzyme. And once it binds to this allosteric site, the enzyme changes conformation such that the active site doesn't allow for the substrate to bind anymore. And the enzyme can't bind to the substrate and can't go on to do its job. So this is a form of inhibition. And then you have the flip of this, which is activation. So the enzyme starts out in a conformation where the substrate can't bind. And so what the activator does is it binds to this allosteric site, which is not the active site. And the enzyme changes conformation, which allows the substrate to bind to the active site, while the activator is bound to the enzyme. So both the substrate and the activator will be bound to the enzyme as like end result, so it can do its job, because once the activator is released, the enzyme will go back into its altered active state where it won't be able to bind to the substrate. So basically just structures function. If you look, the enzyme can't do its function if it's not structured correctly. So that's really all you really need to know. Wow, I can't speak. All you really need to know about allosteric. Laura doesn't like to use it a lot, but if she ever mentions allosteric inhibition and activation, this is what she's talking about. So something she does talk a lot about is post-translational modification. So post, it's in the name, post-translational, it occurs after the protein is made. So this means after tertiary structure. So it's reversible and it's a switch mechanism. What it does is it changes the surface chemistry, it can change the structure, and it can change the function of a protein after it's made. Some examples are methylation, ubiquitination, and acetylation. So this like diagram basically shows you that these little tags are added, which the like keys at the bottom, yellow is phosphorylation, green ubiquitin, blue acetylation. So basically what happens is after the protein is made, so you have this protein X and has a little N and C terminus on it. It's these post-translational modifications are added to it, kind of like these little tags. And then basically based on the tags, so it can be one or the other, it can be multiple. It tells the protein what to do. So after when the code is red, it can either bind to proteins Y and Z, it will move to the nucleus, it'll move to the proteasome, or it will move to the plasma membrane. Those are just some examples. And then this bottom protein with all these little tags on it is an example of P53, which is a tumor suppressor gene, which we'll actually talk about for exam 2. but all you really need to know is that it has these tags, which basically tell it what to do. So the key concept here is that the changes in like the surface chemistry with these modifications will affect the function of the protein. So it tells it what to do. So like one example is ubiquitin mediated proteolysis, which is just a bunch of ubiquitin tags are added to a protein, and it just tells the cell to send the protein to the proteasome for degradation. So these tags just kind of are like markers that tell the protein what to do or where to go. And it's just structure equals function. So nucleotide binding and hydrolysis is somewhat similar. Basically, we talked about this more with like RAN-GAP and RAN-GEF, but GTPases and ATPases, just a quick recap, are categories of enzymes that hydrolyze GTP and ATP respectively. So it's never both, it's one or the other. So ATPases are always ATP and GTPases are always GTP. So they basically just hydrolyze the triphosphate to a diphosphate by losing a phosphate in the process. And this is used to regulate activities and common in the signal transduction pathways that we talked about in 3.1. I don't know if you guys have looked at it because I don't think it's on the exam. It's not on exam 1, but they're used a lot in these pathways, which you'll have to know for exam 2. But basically what this diagram is showing you is that when a GTP binding protein, so this is just a GTPase activity, but it's the same idea for an ATPase. When the protein is bound to GTP, it's on and active, and it can go on and do certain things within the cell. So what the inherent activity, like the activity of the protein is it can hydrolyze GTP by itself. So it loses a phosphate and then now becomes GDP. So then it's off and active. And in order to become active again, it has to lose that GDP molecule and gain a different GTP molecule. So a common misconception that people have is that it just regains a phosphate, but it doesn't. It loses the entire molecule and a new GTP molecule has to diffuse in. But when you're going from GTP to GDP, you are just losing one phosphate. It's pretty simple. But when you're gaining a new GTP, you're getting a whole new molecule. It's a different thing. So that's basically it. An example of ATPases are some helicases. And motor proteins also are examples of ATPases, which I'm sure you guys have heard of, but we definitely go over that more later in the semester. So we move on to protein localization.
Yeah, real quickly, just something I wanted to add, because I saw this question on the discussion forum. Someone asked, can a GTPase work for ATP and vice versa? No, GTPases only work to cleave GTP into GDP, and ATPases are specific for ATP. So you cannot have a GTPase work on an ATP molecule. You'll need an ATPase for that.
Yep.
I saw that question somewhere in the discussion forum. I just wanted to point that out.
Yeah, it's a common question. Jason, for some reason, Jason's another TA for the like regular semesters.
He's my best friend, love of my life.
Yeah, he is the love of your life. He, for some reason, lost his mind at this meme. I like took it out to put another one. He's like, "Why'd you take it out? Put it back." I was like, "Okay, oh my god."
Very protective of his memes.
Yeah, seriously. Okay. I'm doing this one, right?
I'm sorry?
I'm doing this one, right?
Yeah, go for it.
All right. Okay. So protein localization, we started off with signal sequences. So basically, proteins start in eukaryotes. Proteins start translating on a free ribosome in the cytoplasm. So just a quick recap, prokaryotes can translate and transcribe at the same time because there's no nucleus, but in eukaryotes, you transcribe your proteins in the nucleus and then you have to, or the mRNA in the nucleus. So you get that transcript and then you have to transport that mRNA transcript out of the nucleus into the cytosol for translation. So that'll be important for like the trip operon stuff. But for now, just remember that proteins start translating on a free ribosome that's just like floating around in the cytoplasm. That's where they start. And then depending on the signal sequences that it has or does not have, will determine where that protein ends up. So if a protein is localized to the ER anywhere in the endomembrane system, which includes like the Golgi bodies, are the peroxisomes part of the endomembrane system? I don't remember.
Could you say that again?
Are peroxisomes part of the? Yeah, they are. I think they are. Of the endomembrane system.
Yeah, when something needs to be like destroyed, I think.
Yeah.
Okay.
Yeah. So anyways, if the protein is localized anywhere to the endomembrane system or especially the ER, it has to have an ER signal sequence. This is because the ER signal sequence will be recognized, but what's called a signal recognition particle. And it binds to the protein as soon as it's made or as soon as it's translated and ships it off to the ER. So the ER localization signal sequence is located at the very beginning of the protein. So it's near the N-terminus. And basically what happens is as the protein starts translating on this free ribosome in the cytoplasm, the cell will, or the signal recognition particle will notice that it has this ER localization sequence. And as the protein is being translated by this ribosome, it will move, the ribosome will dock to the ER so that as the protein is being translated, it will be immediately put into the ER lumen. And this only occurs if the protein has this ER localization sequence. If it does not have the sequence or has a different one, then the protein will, if it does not have the ER signal sequence or any signal sequence, the protein will not leave the cytosol. It will stay in the cytosol after it has been translated. It will finish translation on that free floating ribosome. The ribosome won't dock to anything. It will just be translated and just do its thing in the cytosol. It won't go anywhere. But if the protein that you're translating needs to be secreted and go outside of the cell to do like its job, it has to go through the entire endomembrane system. So it has to have an ER localization sequence, because in order to get anywhere in the endomembrane system, you have to go through the ER first. And then just a quick side note, once the proteins leave the ER to go through, so the ER is the first step in the endomembrane system, but once the protein leaves it to go through it, like whether it's going to the Golgi's or it's going to leave the cell, it will they rarely come back. So they usually don't go back into the ER. So I know that was kind of a lot, but just some takeaways is that the ER signal sequence is at the beginning. And as the protein's being translated, this SRP will recognize it. And that determines what happens to the ribosome while it's translating and where the protein will end up. So these are some of the trends. So don't memorize the order. So like don't memorize this leucine thing. Just focus on the trends. So if you see that the import into the ER, so the ER signal sequence has a long stretch of these non-polar amino acids near the N-terminus. So you have your N-terminus, you see the N, the little plus sign there, yeah, where Sebastian's cursor is. This long stretch of non-polar amino acids is kind of the tell of a ER signal sequence. Then you have these shit, what's glue? Oh my god, I'm sorry. I forget what is that glutamine? I don't remember. I had them memorized for the MCAT, but I don't remember. But anyways, blue is negative, so acidic. Okay, yeah, so that's glutamic acid. Okay. So then you have these two acidic, and then you have one basic and then an acidic. You don't have to remember like that specific detail. Just know that this long stretch of non-polar amino acids is what's going to signal that you have an ER localization sequence. And then just another trend, like the NLS or nuclear localization sequence is a long stretch of positively charged basic amino acids, these amino acids. So basically you don't have to memorize these. Just remember that the certain trends dictate like where the protein will end up in terms of its sequence. So this is what the SRP is like reading and looking for. Okay.
Wait, sorry, were you done with the slide?
Yeah.
Okay, sweet.
So this is just some of the trends. So like I said, the hydrophobic nonpolar amino acids are near the N-terminus for ER localization. And this is what the SRP binds to the sequence and brings the protein to the ER. If the protein's job is to stay within the ER, it needs a retention sequence. If it doesn't have the sequence, it will just continue to move to the endomembrane system. So if the protein's like final destination is to stay in the ER, it needs a second sequence, which is the ER retention sequence, located near the C-terminus. So it needs both the ER import sequence and the ER retention sequence if the protein is going to stay in the ER. For the mitochondria, just the trend is that it's interspersed positively charged R groups near the N terminus. One thing to note is that once the protein is sent into the mitochondria, the sequence does get cleaved on entry. So I actually don't, Sebastian, do you remember if the sequence gets cleaved for the ER? I don't think it does, right? Because it needs both.
To go to the ER?
Yeah, it doesn't get cleaved when it enters the ER, right?
I honestly don't remember.
I don't think it does, because that would kind of reduce the whole point. Because it needs both to stay in the, okay. It's not on here, so I don't think it's important, but you just have to know that the sequence does get cleaved when it enters the mitochondria. Because once in the mitochondria, it doesn't leave, so there's no need for it to still have this sequence. And then the NLS, like I mentioned, so this one doesn't get cleaved off because this is what important binds do. If you guys remember, nuclear import in a second. So these are just more trends, and exportin, blah, blah, blah. Yeah, also doesn't get cleaved off. These are, so this is just words of what the last slide was showing you. And then just remember that for the cytosol, there is no signal sequence for cytosolic proteins. Any protein that doesn't have a signal sequence. So this is just for reference. This chart is super, super helpful. You can obviously go back and read it, but it basically is just a flow chart of where the proteins will end up and where they won't end up. But it's very, very helpful. Okay, so nuclear import and export, like we mentioned before, use the names. So GEFs are guanine nucleotide exchange factors and GAPs are GTPase activating proteins. So GEFs are involved in exchanging GDP for GTP. So you're making something active by adding that GTP into it. And it does this by binding to RAN, which causes this conformational change for it to increase affinity for GTP and decrease affinity for GDP. GEFs are located in the nucleus and GAPs are located in the cytoplasm. So GAPs hydrolyze GTP to GDP. Like I mentioned, they're the GTPase activating protein. So they're helping with, they have that hydrolyzing activity. So they hydrolyze GTP to GDP. Basically, RAN has its own GTPase activity and GAPs basically just increase the rate of the reaction because it's very, very slow. So these are just the names of the GEFs and the GAPs. And then they kind of make more sense.
Sorry, Rachel, I think that last part got cut off.
Oh, did it? Okay.
Yeah.
I just basically just said that they're just the GEFs are in the nucleus and the GAPs are in the cytoplasm, and it'll make more sense when we look at the actual pictures.
Yeah.
I had a question about the GEFs and the GAPs, but it's like about this unit's problem set. Can I ask that?
Yeah.
Okay. So it's just asking you to go through and it's like explaining to a grad student mentor what would happen if, and then one of them is a loss of function in RAS GAP in the RTK RAS MAPK pathway. But doesn't it not hydrolyze GTP to GDP? So wouldn't there not be a RAS gap?
Okay. So what doesn't hydrolyze it?
Well,
Like-
just in the pathway, doesn't it just go from GDP to GTP?
In the RAS RTK MAPK pathway?
Yeah.
You said there was a loss of function with SOS1.
Is it the SOS1 the RAS GAP?
The RAS GAP. Oh, the RAS GAP.
Because
No.
right, isn't that the RAS GEF?
Wait, sorry, say that again?
Yeah,
Isn't
it's a GEF.
SOS1 the RAS GEF?
Yeah, that's a
Yeah.
GAP.
It's a GEF.
If it lost the function, if it lost its gap function, it can't hydrolyze.
It can't turn off.
Right.
So I would assume it'd just be an overactive RAS signaling, wouldn't it?
Yeah, I'd have to like read the question one more time, and I'd have to like look at the diagram because I personally I'm a very visual person, so I need to like read the question again. I need to look at the diagram, and I could like better describe it that way. If you want to send like either one of us or both of us a screenshot of that problem, we could try to point you in the right direction.
Okay, thank you.
Yeah, no problem.
Sorry, we couldn't give a more comprehensive answer. I'm the same with visual learning and everything.
Yeah, without this, I could never explain this like without.
Oh yeah, no, I know. Yeah, the picture does so much. All right, so nuclear import. We'll just go through the steps real quick, but basically it starts with this cargo protein that has a nuclear localization sequence. This is a little bump out that is there. So what happens is it starts on the cytosol and it has this sequence that's like, hey, you need to go into the nucleus. So what happens is this other protein called important binds to the NLS of the cargo protein, and it carries it through these pores, which are big enough to let the complex in, and it brings it into the nucleus of the cell. Once it's in the nucleus, important needs to let go of the cargo protein, but it can't do that on its own. So what happens is you have this RAN-GEF situation that we talked about before. So GEF helps RAN decrease affinity for GDP and increase affinity for GTP. So basically GEF turns, I almost said GEF turns RAN on, but that sounds wrong. So GEF makes RAN active. And so now GTP is now bound to RAN because it's allowed to diffuse in. RAN basically, I don't know how it changes, it binds to important, and it changes the conformation of important so that important can now let go of the cargo protein. So RAN GTP causes important to let go of the cargo protein. The cargo protein is released into the nucleus and can go off and do its thing. And then now, so RAN GTP is bound to important, and it needs to take important back into the cytosol so the cycle can continue. So it takes it through the pore, and then in order for RAN GTP to let go of important, it has to hydrolyze GTP to GDP. And RAN can do this by itself, but it's very, very slow to the point that the cell wouldn't be able to function if it was RAN on its own. So that's why we have GAP. So GAP increases the rate of RAN's ability to hydrolyze GTP to GDP. And once it does that, RAN can let go of important, and then important can go off and bind to another protein that has an NLS sequence. Do you guys have any questions on nuclear import?
No, I'm all set.
All right, cool.
I have actually a quick question. I saw this on the exam. I just wanted to ask.
Their exam?
No, like previous exams.
Oh, I was like, we have access.
Yeah, no, we don't. What localization sequence do you think important has? Do you think it has an NLS, nuclear export, neither of them? What do you think?
Are you asking me or them?
Anyone?
For important?
Yeah.
I mean, I would imagine, well, the nuclear localization sequence is on the cargo protein, right? Like that's what important binds to, and nothing is binding in turn to important, so I wouldn't think it would have one.
Well, actually anything that goes into the nucleus must have its own nuclear localization sequence. So the pore wouldn't allow, although the pores are very big, they do regulate what goes into the nucleus. They don't just want like anything going inside the nucleus. So the pore will important recognizes this NLS, but the pore will also recognize that important has its own NLS, which allows the whole complex to go in. However, important needs not only a nuclear localization sequence to first get into the nucleus, it also needs to have a nuclear export sequence, an NES, for it to be able to get out, right?
Yeah.
So I saw a question like this on a previous, not this upcoming exam, we haven't seen the exam, but on a previous exam, Laura listed like a bunch of different proteins, like I think RAN-GEF or RAN-GAP was one of them.
Yeah.
It was like, what localization sequences would you expect these guys to have? And the cargo protein, for instance, it would only have an NLS. You don't want the cargo leaving, so it shouldn't have an NES. But for this RAN thing, for important, it would have both because it needs to be able to travel inside as well as outside of the nucleus. Does that make sense?
Yes.
Sweet.
Yeah, if you think of it too, like important is also a protein. It's not like this special like exception. So it needs something to get in and out also. It just doesn't need its own important to bring it into the cell, like or into the nucleus. It does that on its own. All right, so export is the reverse basically. So we start with a protein in the nucleus that has this NES sequence, so the nuclear export sequence. And it needs to get out of the nucleus, but it does this by binding to not only exportin, but in order for exportin to bind to the cargo protein, it needs to also be bound to RAN-GTP. So the same thing happens. GEF helps RAN become active by allowing, by changing its conformation and decreasing its affinity for GDP and increasing its affinity for GTP. So RAN-GTP is now active thanks to GEF. It binds to exportin, sorry, and exportin binds to the cargo protein at its nuclear export sequence. So now you have this complex of 3 different like proteins, enzymes, whatever, and they can exit the pore. And once they're in the cytosol, the same thing needs to happen for RAN-GTP to let go of the complex. So GAP helps RAN hydrolyze GTP to GDP, and then it can let go of exportin and the cargo protein at the same time. So the cargo is released into the cytosol, it can go on and do its thing. And then exportin will re-enter the nucleus by itself and can restart the whole cycle. Just a quick side note, fully folded proteins can move in and out of the nucleus because of these pores. They're very big, so sometimes you might ask a question if, I don't know, if it's asking if a protein can go in fully folded, it can. Something that, an example of it not, a protein not being able to enter an organelle fully folded would be the mitochondria. It can't enter fully folded into the mitochondria. It has to be unfolded into like the individual polypeptide strand, like just to primary structure, and then it's like sorted into the mitochondria and then it refolds once it's inside. That's not the case for the nucleus. It can enter fully folded. And the signal sequence for both export and import is not cleaved once it does its job. So those are just some side notes. We have some practice questions. Oh my God. Okay. Okay, so we'll go through these pretty quickly. If a protein has both an ER localization sequence and a mitochondrial localization sequence, where will the protein end up? What do you guys think?
Would it be the ER?
Why do you think so?
Because it's being translated by the ribosome, and that sequence, I know you mentioned it's very early. And so when it's translated, then that would bind and it would feed it into the ER, though I don't know how the mitochondrial localization sequence works, but I would guess the ER.
Yeah, so that's basically the answer. It's based on the order. So the ER localization sequence is the like earliest sequence, so it will be read first if a protein has both. And then because the mitochondrial sequence comes later on in the protein, but once the protein is in the ER, it really won't exit it. So the mitochondrial localization sequence really doesn't have a function here. Does that make sense?
Yeah.
Okay, cool. All right, number 2, if a protein has an ER retention sequence, a nuclear localization sequence, and a Golgi localization sequence, where will the protein end up?
What? Retention sequence?
I would say the ER.
Why do you say
Oh,
that?
wait, it doesn't have an ER localization sequence, though. So then it would be the nucleus.
Exactly. Good catch. So, right, it needs that localization sequence to even end up in the ER. The retention sequence won't do anything if it doesn't have it. And the Golgi is a part of the endomembrane system, so it can't end up there because it can't enter the endomembrane system through the ER. So you're right. So it has the only sequence that would let the protein do anything is the nuclear one, so it will end up in the nucleus. Right. Number 3, if a protein has an ER localization sequence, where will the protein end up?
Really?
No.
Oh,
It'll be secreted out of the cell.
Right, because it has this localization sequence, so it will enter the endomembrane system. There's no mention of a retention sequence, so it'll just go all the way through. All right, 4, true or false, the protein enters the nucleus fully folded.
Aren't the pores large enough that it can enter unfolded?
It enters folded, yeah.
Yeah.
The pores are big enough that the protein will enter fully folded. Okay. 5, the signal sequence is cleaved off once it enters the nucleus. Is that true or false?
I believe that's true.
So it's actually false because the signal sequence does not get cleaved off in terms of the like entering or exiting the nucleus. The
Oh, right.
protein will keep it, right? The only times it is cleaved off is if the protein is entering the ER or the mitochondria. That's when the signal sequence is cleaved off. Okay, number 6, true or false? The protein enters the mitochondria fully folded. So we talked about the nucleus. It can enter the nucleus fully folded because it has those pores, but what about the mitochondria? Mitochondria does not have any pores.
So that would be false.
Right, yeah, it's the mitochondria, I mean, it does have pores, but they're very, very small. So the protein will just go back to its primary structure. It'll be sequestered in, and then it will refold once it's back inside of the mitochondria. And then the last one, where will a protein with no localization sequence like at all end up? Doesn't have anything, any zip
The
code.
cytosol.
Good job. Good job.
nice.
Is this you, Sebastian? This is you.
Okay, I love this picture, by the way.
Oh, I know.
Okay, gene
I think this-
expression. Oh, these are definitely
this-
cats.
Are
Yeah.
these Jason's cats?
Yeah, that's Cloud.
Oh,
Okay, this is a good like question break, I guess. Do you guys have any questions so far about anything we've talked about?
we're almost done. You guys can do it.
Yeah, we're in that final stretch. Okay, let's do this. What is that?
Oh my God, this happened while we were making it, and I somehow accidentally put this transition in, and I didn't know how to delete it, so it's just there.
That's so funny. La la
Yep.
la. Okay.
La, la.
So gene expression regulation, as the name implies, the cell wants to be very careful with, or it wants to regulate very carefully how it expresses its genes, how it makes proteins. So one way that it could do that is, oh wait, no, this is something else. Okay. Oh, this is, wait a minute, what does it say?
This is like transcription factors,
Okay.
mediated transcriptional regulation stuff.
Okay, sweet. So as I was saying, the cell wants to be careful with how it regulates its proteins, how it expresses its genes. One way that it does that is through transcription factors and the RNA polymerase itself. So the RNA polymerase, obviously it catalyzes nucleotides being placed onto your template, but before it could do that, it needs the help of transcription factors. So as the name implies, a transcription factor is just a factor that regulates transcription. So you could have positive regulators, you could have negative regulators. Some transcription factors promote polymerization of your of whatever strand you're synthesizing. Other of those repress that. That would be a negative transcription factors. The RNA polymerase can't do it on its own. It needs the help of a transcription factor to bind onto the DNA or the RNA template or whatever, and then start transcribing. So again, the RNA polymerase, of course, does the transcribing, but it needs the help of transcription factors. And depending on what those transcription factors are, will tell you if you have like a positive or if you have a promotion of transcription or a suppression of transcription. So that's one level of regulation. Another one is the chromatin packaging. This is, this is, so your DNA could condense itself or it could decondense. If, like, let's say the cell wants some kind of protein produced, it would decondense the stretch of the DNA that codes for that particular protein. If it's no longer needed, one way that the cell, again, regulates its transcription or its gene expression is it could condense the DNA to where the RNA polymerase and transcription factors cannot touch that region of the DNA. So again, the cell's very careful with what genes it exposes to be transcribed and what genes it wants to conceal. So when it needs a gene expressed, it will decondense that region to allow the polymerase and the transcription factors to get in there and start transcribing. If it does not need those genes expressed, it will condense and it won't allow anything to bind onto the DNA. RNA could be modified. So after the DNA, so going back to like central dogma, after your DNA polymerase has transcribed that stretch, eukaryotes need to process this RNA. Bacteria don't have any RNA processing because they translate their transcript at the same time that it's being transcribed. Transcription and translation happen simultaneously with bacteria, but in eukaryotic cells, as you guys know, everything is transcribed in the nucleus, but that mRNA transcript has to be shipped out. So before that mRNA could be, or that RNA transcript could be shipped out into the cytosol to be translated, that piece of RNA that the polymerase just transcribed needs to be modified. It modifies it for protection to protect the mRNA transcript from being degraded inside the nucleus. And it also adds stability. So one thing that it does is it adds a 5' cap to your RNA transcript. It adds the poly-A-tail at the end of it. I think the poly-A-tail had like some, the poly-A-tail does, it has some role with nuclear export. So it's kind of like a signal. It's kind of like a signal sequence that tells you this piece of genetic material needs to be shipped out of the nucleus. So again, one modification that it does is the addition of the 5' cap, the 3' poly-A-tail. Another thing that it needs to do is it needs to splice out your introns. So introns are just a stretch of like non-coding regions. They don't code for anything. Obviously, you really need to get these introns because they don't code for any protein. You really need to get them cut out before it could be shipped out of the nucleus. Because of course, if your ribosome starts translating this with the introns here, you have nucleotides that don't code anything. So your protein is going to come out with a bunch of amino acids that it doesn't need. That's going to mess up the folding. That's going to mess up the function of your protein. So it's really important that you get your introns spliced out. It does this with a spliceosome, which is an enzyme specific to cutting out introns. Alternative splicing. What's that? This mechanism is called alternative splicing. Oh, okay. Okay, yeah. Alternative splicing is just the regulatory control for cutting out introns. Of course, you don't want to cut out your exons. Your exons do code for the protein. So you don't want those cut out. You want it alternatively spliced. So wherever your introns are. And then finally, you'll have your mature mRNA transcript that'll be ready to be shipped out of the nucleus and ready to be translated. Okay, and here's a really good chart about some of the differences between your eukaryotes and prokaryotes. Again, it's really important that you keep those differences or that you bear in mind the differences between eukaryotes and prokaryotes. Here are just some of them. Okay, any questions so far about any of this?
No.
Sweet. Okay, going back to what I mentioned before about transcription factors. So transcription factors is one way that the cell could regulate its gene expression. So again, the cell is very careful with what genes it expresses and which genes it does not express. So if the cell does not want this stretch or this gene transcribed, it will probably have a bunch of negative regulatory transcription factors, which will suppress the transcription of this particular gene. But if the cell does want that gene to be transcribed, it will most likely have positive regulators to help the DNA polymerase first bind and also transcribe. Here are your general transcription factors. As the name implies, they're just general. They're not for like specific regions of the DNA. You could kind of use these guys for any part of the DNA or like just general regions of the DNA. A regulatory transcription factor would be if you want like a very specific kind of gene transcribed, then you would most likely see one of these. General transcription factors, again, they're just used for generally around the DNA. Bacteria have sigma factors, which I'll talk about in a minute. Eukaryotes have basal transcription factors, and again, those are just your general transcription factors. Those transcription factors work generally all around the DNA. They're not very signal specific. Whereas the regulatory transcription factors, these are a little more specific to a particular kind of gene, most likely with like housekeeping genes, pretty important genes. Okay, any questions about these? Okay, why can prokaryotes simultaneously transcribe and translate the mRNA, but eukaryotes cannot? Why do you think?
Is it because of histones?
Yeah,
They
prokaryotes, I know they don't have, well, not exactly, but yes, they don't have that level of regulation. Because they don't have histones, they don't really do the, they have some level of transcriptional control, but not the way the eukaryotes do. So prokaryotes don't condense and decondense their DNA. They have different ways of doing that, but you're right, they don't have histones. But the main reason why they're able to transcribe and translate at the same time is because they lack a nuclear envelope. Obviously, eukaryotes, we do have a nucleus, so you do need some kind of mechanism or some kind of regulatory process to get that mRNA from the nucleus out of the nucleus. So that's why you need to process it a little bit more. You need to add that 5' cap, that 3' poly A tail, splice out the introns. That's all preparing it to be shipped out of the nucleus. But for prokaryotes, they don't have a nucleus. Pretty much everything is just the cytosol. So they are able to translate at the same time that the cell is transcribing straight off of the DNA. Does that make sense?
Yeah.
Okay. True or false? RNA polymerase can bind to the DNA on its own. This is like 100 times. What's the next one? For the sake of time, let's just go to number 3. The answer is, of course, false. The RNA polymerase cannot bind to the DNA on its own. It needs the help of transcription factors to bind. Okay, so which of the following is not true regarding gene expression in prokaryotes? Prokaryotes regulate mRNA localization, regulate mRNA degradation, they package their DNA into chromatin, or prokaryotes regulate the rate of translation. So which one of these is not true regarding prokaryotes? We kind of mentioned this a little earlier.
See?
Yeah, exactly. They don't have, or they don't condense their DNA into chromatin the way we do. They don't have histones, so they don't have that level of regulation. So wait, I'm sorry, did you say C or D?
C.
Oh, yes. Okay, yeah, then you're right. Yes, it is C. Name some examples of post-translational modification. Honestly, I couldn't even tell you. The only one that comes to mind is the ubiquitin tag. So post-translational is after your protein has been made. Hence the name post-translation.
Was this like the methylation and the acetylation?
Yes, exactly.
Would phosphorylation also be post-translational modification?
That's a good question for Rachel.
Yes.
You think-
So phosphorylation, acetylation, dephosphorylation, ubiquitination, and protein degradation, and methylation, those are all post-translational.
Yeah, awesome. I know, again, like on a previous exam, I've seen questions that it gives you like a whole bunch of scenarios, like introns being sliced out. What kind of regulation is this? And that would be a level of transcriptional control or post-transcriptional control. It's post-transcriptional because your transcript has already been transcribed. Splicing out the introns is post-transcriptional modification. If you see something that's like, well, what about decondensation of the DNA? That would be transcriptional control. So that's another kind of question that Laura likes to add in. So just in case you see anything like that, you're prepared. Okay, is alternative splicing okay, perfect. Is alternative splicing an example of post-translational modification? What do you think? Wow, I didn't even read that.
No,
Why do you say that?
because it's, I mean, alternative splicing is in the eukaryotic cell in the nucleus when it's to cut out the introns in the RNA. So the protein has not been translated yet.
Exactly. Yeah.
So it'd be
Post-translational.
transcriptional.
Yes. Yes, exactly. Post-translational implies that the protein is already synthesized, it's already made. In that case, yeah, you could add tags, acetylation, methylation, ubiquitation, whatever. But alternative splicing, getting out the introns, yeah, that would be post-transcriptional. Sweet. Okay, the trip operon and RNA interference. This is like the bane of everyone's existence, but I'll try my best to like simplify this. So this is
section. Sorry, I'm switching to the last section.
Yeah, nice. Okay, so prokaryotes have these things called operons, where unlike eukaryotes, we don't have this kind of system where I just lost my train of thought. Eukaryotes generally have monocystic kind of regulation of transcription. So one gene codes for one protein, whereas here you could have multiple genes coding for multiple proteins. And that's essentially what an operon does. An operon is just, you have one promoter that can transcribe multiple genes that code for multiple different proteins. And this is something that's pretty unique to prokaryotes. So this diagram, okay, all of these diagrams are gonna, sorry, I forgot the puppy break. Okay, anyway, yeah, the diagrams that you'll find on like Google Images vary a little bit, but essentially you have the main parts that you should bear in mind is the promoter region. I'll talk about what that is. The operator region. Between the operator and the structural genes, you have the leader strand. That's where like most of the confusion comes from in this section, but I'll get to that later. So right before you get to the structural genes, you have the leader sequence, which I'll talk about in a minute. But again, the main parts, the main anatomy of the operon is the promoter region. The promoter region is where your polymerase binds to. It's where it hooks onto. The operator region is the region that tells the polymerase, can it proceed with transcription or can it not proceed with transcription? The leader sequence, which it doesn't really specify in this diagram, but there's a leader sequence like somewhere right here. The leader sequence is involved in another level of regulation, which I'll talk about in a second. Okay, operons come in an inducible, they have an inducible form or a repressible form. This trip operon is a form of, it's a type of repressible operon. It's repressible because the operator is essentially always on unless it is not on. I feel like explaining this with an example is probably going to be a little more helpful. Okay, so there's a bunch of different operons, trick operon, lac operon, there's a bunch. The one that we're going to focus on in this class is the trip operon. So there's two levels of regulation with the trip operon. The first level is pretty straightforward. It's pretty easy. The second one is where you get like a little more, it gets a little more confusing. But the first level of regulation, let's just focus on that. In the first level of regulation, the cell just asks itself, is there a lot of tryptophan or is there very little tryptophan? if there's low levels of tryptophan, the repressor is going to be off. Okay, I'm going to be doing my explaining on this slide. So if there's high level of tryptophan, it's likely the tryptophan is going to diffuse into the repressor. If it diffuses into the repressor, it's going to bind onto the operator. Now that the repressor is bound to the operator, let's say your polymerase has, oh no, actually, sorry, forget that. If your repressor is bound to the operator, it is blocking the RNA polymerase from binding to the promoter. Remember, this promoter is just the stretch of the operon that tells the polymerase where to bind. But if the repressor is connected to the operator, if the repressor is bound, it is blocking the site where the RNA polymerase can bind to, so the polymerase cannot bind and no transcription can happen. So that's the first level of regulation. The first level is where the cell just asks itself, is there a lot of tryptophan or is there not? If there's a lot of tryptophan, the tryptophan will connect to the repressor, it'll connect to the operator, no transcription will happen. If there is no tryptophan available, if there's very, very little tryptophan, no tryptophan will be connected to the repressor. The repressor will not be connected to the operator. And because the repressor is not connected to the operator, the RNA polymerase is free to land on the polymerase, or excuse me, on the promoter region with the help of transcription factors, and it's allowed to transcribe. Okay, so any questions about this first level of regulation? It's pretty straightforward. Okay, so one more time, just to simplify, low tryptophan, there will be transcription. High tryptophan, there will not be transcription. Okay, the second level is when you talk about the leader strand. So this is a little more, it does get a little trickier. So this is your second level of regulation. In this case, this would be where if you have like a medium amount of tryptophan. What is this? Okay, so let's say the cell is very, very starved of tryptophan. Oh, wait a minute. Okay, yeah. Let's say the cell is very, very starved of tryptophan. There's no tryptophan to be available. If there's no tryptophan available, nothing will connect to the repressor. The repressor will not connect to the operator region. If it's not connected to the operator region, the RNA polymerase could bind and it could start transcribing. So if that's the case, the RNA polymerase is going to connect itself somewhere here, and it's going to start transcribing. It's going to continue transcribing until it gets right here, about this region, the transcription pause. Okay, remember that in prokaryotes, transcription and translation happen at the same time. So as the RNA polymerase is transcribing the transcript, the ribosome is right behind it. The ribosome is trailing the polymerase right behind, and it's translating at the same time. Remember that in this cell is very starved of tryptophan. So what's going to happen is your ribosome is going to connect to the transcript. It's going to start translating like la la la, and then it's going to reach like this region right here. If the ribosome reaches this region that codes for 2 tryptophans, the ribosome needs tRNA tryptophan. But mind you, this cell is completely starved of tryptophan. So if the cell is starved of tryptophan, the ribosome cannot proceed from this part. It's going to stop right here. It's going to stop around region 1. So because the because the ribosome has stopped on region 1, that allows for region 2 and 3 to hairpin. Because these 2 regions have hairpin loop, this is a non-termination sequence. So it's going to pause for a little bit for the cell to eventually like muster up whatever tryptophan it has. And once it does find the tRNA tryptophan available for the polymerase to proceed, it's going to proceed and it's going to destroy this hairpin loop, and it's going to make the full mRNA transcript. Does that make sense so far?
Yes.
Sweet. Okay. You might see a question, you might run into a question that's something like, what if you have like a medium amount of tryptophan? So you don't have enough tryptophan where it could bind to the repressor and prevent the polymerase from binding. So you don't have enough to let the repressor repress. But let's say you have a medium amount. In that case, the RNA polymerase, it's going to start transcribing, it's going to do its thing, and again, it's going to pause somewhere right here. The ribosome is going to begin translating. It's going to translate, and it's going to reach this region. So remember in the previous example where tryptophan was completely starved, the ribosome was forced to stop here. The ribosome was forced to stop because it doesn't have the TRP anticodons to place right here. So it's forced to stop. But in this situation, right now, in this situation, this is where the cell has a medium amount of tryptophan. So because it's not completely starved, because there still is some amount of tryptophan, the ribosome is going to be able to add the tryptophan codons here. So it's going to add the tryptophan codons, and it's going to continue going until it reaches the stop codon region. Once the ribosome stops here at the stop codon, notice that it stopped on region 2. If it stopped on region 2, it's not going to be able to bind with region 3 because there's a giant ribosome sitting right here. However, region 3 will be able to hairpin loop with region 4. If that happens, the transcript is kind of, it's pretty much pulled out of the reading frame from the RNA polymerase. So the RNA polymerase is forced to stop. It doesn't have anything to transcribe anymore, so it's just like free floating. So again, because the ribosome was paused right here, right on region 2 on the stop codon, it prevents 2 and 3 from binding, but 3 and 4 can still bind. Because 3 and 4 bind, it yanks that RNA transcript out of the reading frame. So it cannot continue with transcription. And what ends up happening is you have a short transcript. You have an attenuated transcript. Does that make sense? This is arguably like one of the most confusing parts of this class. So don't worry if this is still like mind-boggling to you. I promise practicing it will.
It mostly makes sense. My one question is, what is the connection between the amount of tryptophan in the cell and the amount of tryptophan tRNA? Like,
Okay.
if we have low tryptophan, does that mean something about the amount of tryptophan tRNA?
Yeah. Yeah. If you have low tryptophan, you're gonna have low tRNA tryptophan. Laura doesn't usually mention the tRNA tryptophan concentration. When she would usually just say like a medium amount. So Laura's terminology is usually if there's high tryptophan. If there's high tryptophan, then the repressor is almost certainly bound. If there's low tryptophan, so if she uses the words like if there's very, very low tryptophan, then the repressor is off, there's no tRNA tryptophan, and you're gonna get the full mRNA transcript. But if she says something like there's a medium amount of tryptophan, if that's the case, then you don't have enough tryptophan to make the repressor bind to the operator, oh God, but you do have enough tryptophan where attenuation will occur, where you'll end up with the short mRNA transcript. Does that answer your question?
Yeah, thanks.
Okay, no problem. Okay, so what happens when there's a high concentration of tryptophan? Pretty straightforward.
Repressor binds.
Exactly, repressor's bound, no transcription happens. Okay, let's say region 4 is deleted. What's gonna be the size of the mRNA transcript, and why do you think? So if we delete region 4, what do you think might happen?
It would be a full transcript.
Yeah, every single time. Even if you have a low or a medium amount, if you delete region 4, you can't get this 3-4 hairpin loop to form. And if it can't form, you can't get an attenuated mRNA transcript. So yes, the full transcript will be made. Okay, let's say I know you hate this question, Rachel, but let's say you mutate the UGG that codes for tryptophan sequence, and you mutated it to a cysteine codon instead. So this part. And let's say that there's plenty of tRNA cysteine around. What's going to be the size of the RNA transcript? If you mutate this tryptophan region, to cysteine, and you have a lot of cysteine anticodon around, what do you think the size of the mRNA transcript will be?
Full. I forget if it goes to the I know it would go to the stop codon. And so does that mean that that means the 3-4 loop would form, so it would be terminated.
Yes, exactly. If you mutate this to cysteine and you have a bunch of cysteine floating around in the cell, the ribosome is going to have it's going to have no problem adding the anticodons to this part. So it's going to continue and it's going to rest here on region 2. If it rests on region 2, then 3 and 4 will hairpin loop, and if they hairpin loop, then you get the short mRNA transcript. That's exactly right. Sweet. Okay. True or false? Attenuation will only occur when tryptophan levels are high. What do you think?
False.
Why do you think so?
Well, if tryptophan levels were high, the repressor would bind, and so attenuation would probably not need to occur.
Exactly. This is a very, very typical LoRa kind of question. If your tryptophan levels are high, this second level of regulation, this attenuation, whatever, that's not even going to occur. If tryptophan level is high, tryptophan is going to be bound to the repressor is going to be bound to the operator, and your RNA polymerase isn't even going to be able to bind to the DNA. So yeah, this question is false. Sweet. Okay. Any questions about anything before I jump into RNAi? You want to take this, Rachel? Just kidding. Okay, so RNA interference is just a way for the cell to control, to regulate its gene expression. It's another way of gene expression regulation. So the two main kinds of RNA interferences that we'll talk about is miRNA, microRNA, and small interfering RNA, siRNA. Again, it's important to know the differences and the similarities between the two, which I'll definitely go into. Here's some pretty cool facts about it. Actually, the guy who won the Nobel Prize for, I think he didn't discover, but I think he had some involvement with siRNA. I think he was one of like the first researchers to develop it, or to I guess get familiar with that technology. He actually went to UMass Med School, and he's a professor there at the medical school in Worcester. Yeah. Anyway, okay. So miRNA, microRNA. Hold on, let me just get a gauge. Okay, sweet. MicroRNA is this kind of RNA interference is the one that our cells do naturally. So there's mi, which is the ones that that our cells do naturally. siRNA is the one that has to be exogenously like inserted into the cells. So that's one of the differences, but I'll get to that later. So in the miRNA pathway, that all starts in the nucleus. So again, let's say there's some mRNA transcript that the cell really wants to get rid of. What it's going to do is it's going to make a really, really long transcript called the pri-miRNA. This diagram is wrong, by the way. This is supposed to say pri, P-R-I, not pre-miRNA. But anyway, what it's going to do is it's going to transcribe this really, really, really long piece of RNA called pri-miRNA. There's an enzyme in the nucleus called DROSHA that cuts that really long piece of miRNA, excuse me, that really long pri-miRNA into pre-miRNA. Pre-miRNA is then exported out of the nucleus, where another enzyme in the cytosol called dicer cuts it even further into these miRNA duplexes. There is another protein called Argonaute that finds these duplexes and it connects it to it. When Argonaute binds to the miRNA duplex, that's called the RISC complex. What the RISC complex then does is so again, these are RNA duplexes. So one one strand of this duplex has the exact same sequence as the target mRNA, and the other strand has a complementary sequence to the miRNA. So RISC is trying to get this miRNA duplex, or it's trying to get the miRNA to have a complementary binding sequence to the target mRNA. So what the RISC complex is going to do, it's going to unwind the duplex. It's going to discard the strand that has exact similarity, because it doesn't want that one. There's no way for it to bind if it has the exact same sequence. It's going to, again, separate the strands, get rid of the strand that has the exact same sequence, and it's just going to hold on to the strand that has complementary sequence to the target mRNA. So that's exactly what it does. The exact match is less common. Argonaut cleaves mRNA. Degraded partial match more common. Yeah. Okay. So any questions about the miRNA pathway? Another thing to point out is that miRNA only has like a partial similarity to the target mRNA. It doesn't have an exact complementary sequence, unlike siRNA. In siRNA, the process is almost identical, except it does not begin in the nucleus. So in miRNA, all that stuff begins in the nucleus. Here, like I mentioned before, with small interfering RNA, it has to be exogenously inserted into the cell with like a needle, like some kind of injection. And it could come in the form of double-stranded RNA, or it could be a small hairpin RNA. Whatever the case may be, the following steps are pretty much the same. Dicer recognizes this double-stranded RNA, or the small hairpin RNA. It's going to cut it into the duplexes. Argonaut is going to recognize that duplex, and it's going to hold onto it, which forms the RISC complex. The RISC complex is then going to separate the strands. It's going to keep the strand that has complementary sequence to the target mRNA, and it's going to discard the one that has exact similarity to the to the target mRNA. So I mentioned before that one of the differences between si and miRNA is that siRNA begins in the cytosol, whereas the miRNA begins in the nucleus. Another difference is that, as I mentioned before, miRNA has partial complementation to the target mRNA, whereas siRNA has a 100% complementation to the target mRNA. And the reason for that, because it has a 100% complementation sequence, is because siRNA is predominantly used for research purposes. So if you're a researcher and you're trying to do like a knockout experiment and you want to get rid of an mRNA to see how that phenotypically affects the cell, you know what the sequence is of the mRNA that you want to get rid of. So because you know the sequence of the mRNA, you'll know exactly what the complementation sequence is to target that mRNA. So again, because siRNA is something that you kind of develop in a lab, that's how you're able to have 100% complementation, because you already know going into the experiment what complementation you want to get rid of. Does that make sense? Okay, what advantage of miRNA? Okay, so I kind of touched upon this. Just to save us time, I'll just articulate these, or actually, yeah, I'll just give it to you guys. Do you guys have any ideas as to why, like what advantage miRNA has not been 100% complementary, unlike siRNA?
It can act on a great number of RNA that are maybe involved in like similar processes, so it can work more on like pathways rather than just very specific individual RNA.
Yeah, exactly. By not having 100% complementation, like you said, RNA interference can be done on a more broad range of mRNA transcripts. It's not just specific for like one very specific transcript. It could work on a bunch of different transcripts that may have like similarities between the two. And true or false, Drosha turns pri-mRNA into pre-mRNA in the cytoplasm. This is supposed to be pre-miRNA, by the way. Pri and pre-miRNA.
False.
Let me check because I don't know. Yes, it is false because Drosha only functions in the nucleus, not the cytoplasm. Sweet. Okay. Nice. All right, y'all. How are you feeling about this exam Saturday?
Now that I know it's open notes, I'm feeling way better.
Yeah, I'm pretty
So,
sure. Yeah, I don't think Laura's gonna like make this stuff. There's really no way to even enforce that, you know. So yeah,
Yeah.
I think yeah, your notes.
She just
will
doesn't
be
want you to
written-
work with other people.
Exactly.
But
Yeah.
honestly, even then, we can't really monitor that either. But
Yeah, I mean, we do have the similarity score thing, and sometimes it is pretty obvious, especially when the class is not as small. Like I would oftentimes notice when people worked with each other in a class of like 200 people. Not always, but sometimes it was a little obvious. In a class of like 17 people or something, it could be a lot more obvious. But yeah, that's the only thing. You're welcome to use your notes, internet, whatever it may be. Just not humans. Oh, and if you do have a question about like wording or something like during the exam, you can of course email me, Rachel, or Laura. The only thing that we ask is if you are going to email for like a clarification question or something, be sure to tag all of us in it. Like if you're emailing me or Rachel, be sure to just tag Laura in it as well. Yeah, actually, okay, so if you're emailing Rachel with a question, for example, be sure to tag Laura in it as well. You don't have to tag me, but please do tag Laura in it, because she could obviously help you out better. She can most likely help you out better than we can. So yeah. Those instructions will be posted at the beginning of the exam anyway, so in case you forget or whatever, it'll be there. All right, y'all. So that's all we have for you.
good luck. You're
Yeah.
gonna do great.
oh yeah, thanks so much.
Thanks.
Yeah.
This was very helpful.
Anytime.
Good, yay.
All right, have a good one,
Have a
guys.
good one, guys.
See ya.